So this is the Unit 2 review for AP Biology continued, Part 2. And so here we go. So phospholipids have both hydrophilic and hydrophobic regions. The hydrophilic phosphate regions of the phospholipids are oriented toward the aqueous external and internal environments, while the hydrophobic fatty acid regions face each other within the interior of the membrane. So when we look at what this is talking about, this essential knowledge, basically you can look at a phospholipid molecule and see it has a polar region, which is hydrophilic and attracted to water, as well as a nonpolar region, which is hydrophobic and repels water. So when we talk about a lipid bilayer, the reason it is a bilayer or two layers of these phospholipids is because the inside of the cell is aqueous or a watery environment, and the outside of the cell is also aqueous or a watery environment. And therefore, you find the hydrophilic water-loving regions of the phospholipids attracted to that area, while the hydrophobic water-fearing regions of the fatty acid tails will be facing each other. You would not find a single layer of phospholipids as a cell membrane unless like the inside of the cell was oil or something, or like a non-aqueous inside. Okay. So embedded proteins can be hydrophilic with charged and polar side groups, or hydrophobic with nonpolar side groups. So the beauty of proteins is that their monomer has different, or can have different chemical properties, right? So a protein, some amino acids can be hydrophilic, while some can be hydrophobic, or polar or nonpolar, and that, or those amino acids will play a role when they are, like how you find it, I guess, within the membrane. So the hydrophobic part of the protein is the part that you find within that fatty acid region. So you can imagine that the amino acids like on the outside of that protein will be hydrophobic, because if they were hydrophilic or polar, they'd repel in that region. And then the outside parts, the part of the protein that you find in the extracellular or outside of the cell, or intracellular regions or inside the cell, would be made out of your polar or hydrophilic amino acids. So now this like dual properties of these proteins is also what helps anchor the transmembrane or integral proteins within that lipid bilayer, so they don't just like pop out or something. And when we look at cell membranes, they really are a mosaic. So when you hear the word mosaic, it's meaning that it's made of a whole bunch of different molecules, right? So you have your transmembrane proteins, which are orange here. You have your phospholipids, but there's also steroid molecules such as cholesterol. So cholesterol is a lipid, and it's found embedded within the cell membrane. It acts as like a temperature buffer to help with the fluidity of the membrane. It prevents the membrane from becoming too fluid, but also prevents the membrane from becoming too rigid. Then you have these like carbohydrate chains that come off the surface of the cell. So if the it's called an oligosaccharide. You know how you hear like mono or disaccharide is like one or two sugars, and then a polysaccharide is many? Well, here you have a few. So these oligo, like a few carbohydrates or monosaccharides joined together, you have these chains. The prefix glyco stands for like carbohydrate, like glycogen or glycolysis or something. So you have this glycolipid where the carbohydrate chain would be attached to the fatty acids, and that's what anchors it into the membrane. Or you could have a glycoprotein, which is a strand of carbohydrates, but attached to a transmembrane protein. So these carbohydrate chains are often used for like cell identification or cell communication within organisms. So that is the beautiful mosaic of a cell membrane. And you often hear, I'm sure it's in the standard coming up, but it's described as the fluid mosaic model because it also is fluid and flexible and moving. So this structure of the cell membrane, though, does result in selective permeability. The fact that that lipid bilayer has that nonpolar center does prevent some molecules from entering by simple diffusion. So molecules that will be charged, like ions, or polar molecules like ammonia, for example, will not be able to just diffuse right through the lipid bilayer. So therefore, the lipid bilayer is selectively permeable. Some things can cross, like your small nonpolar molecules like carbon dioxide and oxygen, whereas your polar molecules and your ions, and your molecules that are too large, like macromolecules like carbohydrates or proteins, they cannot just diffuse through that membrane. Now, water is a special case that we'll talk about. And so cell membranes, they do separate the internal from the external environment of the cell. Okay. And here's that fluid mosaic model I was talking about. So that selective permeability is due to the fluid mosaic model. So it includes both the protein channels as well as that lipid bilayer. And small molecules, including nitrogen, oxygen, and carbon dioxide, freely pass through or cross the membrane due to their nonpolar nature. And the hydrophilic substances, such as large polar molecules and ions, move across the membrane through protein channels. So here you can see in this little GIF, the blue are like small nonpolar molecules that can diffuse through, where the green are larger and can't. So polar uncharged molecules, including water, can pass through in small amounts. Now this seems like counter to what I just said, but if we think about a cell, how a cell is surrounded both inside and outside in water, and it's a fluid mosaic model, meaning that the cell can move. Now think about it, some water molecules are going to be able to slip through that lipid bilayer. However, it's only going to be a small amount of them. So when we talk about osmosis in a little bit, water is mainly diffusing through proteins called aquaporins that allow for rapid movement of water into or out of a cell. So that aquaporin creates a little channel for the water to flow in or out, depending on solute concentration. Now, cell walls provide a structural boundary as well as a permeability barrier for some substances to the internal environments of a cell. So over here on the left, this gray picture is like an electron micrograph of cellulose, of the cell wall of a plant. You can see how water and other molecules can still move through that. It's not like a brick wall. And then in our prokaryotes, so I do want to mention though, when we talk about cell walls, the cell walls serve a lot of the same functions like structure or support, but they are made of different things, different types of carbohydrates. So in plants, your cell wall is going to be made of cellulose, whereas in bacteria, it's going to be made of peptoglycan. And in fungi, it'll be made of chitin. So when we look at these cell walls, they are structural and they're not flexible. So how an animal cell can do endo or exocytosis, you would not find that in a plant cell. In a plant cell, if molecules need to transfer from or flow or move from one cell to another, plant cells have these channels or gaps called plasmodesmata between two cells that is like a connection of two cell cytoplasm, so ions and signaling molecules can move through from one cell to the next. So here's what I was mentioning. So cell walls of plants, prokaryotes, and fungi, they are made of carbohydrates, but the type of carbohydrate is different. So here in bacteria, you can see this word peptoglycan. That is the name of the carbohydrate chains, the polysaccharide that compose that cell wall. In bacteria, if you're going into the medical field, you do have bacteria can be divided into two groups. You have your gram-positive and your gram-negative bacteria, and that really comes down to how their cell walls are made. And this plays a role when prescribing antibiotics. Some antibiotics will target gram-positive, while others will target gram-negative. And the cell wall of fungi are made of chitin. So this chitin right here is the cell wall of fungus. And it is also the same polysaccharide you find in the exoskeletons of lobsters and crabs and roly-polies. Okay, so on to passive transport. So passive transport is, as the name implies, passive. It doesn't require any energy and is the net movement. So well, let me finish the thing. Is the net movement of molecules from high concentration to low concentration without the direct input of metabolic energy. So when we talk about net movement, I want to point out that diffusion is due to the random collision of molecules, like colliding into each other and other, not just each other, but other molecules, other surfaces, etc. And they don't like see an area of low concentration and be like, oh, I need to go there. Rather, due to all these random collisions, the overall net movement is from where there's more of them to where there's less. They basically are spreading out from high concentration to low concentration. And it just so happens that because maybe it's like oxygen or carbon dioxide, that they're nonpolar and they can diffuse right through the membrane. I also really like this GIF because you can see how that is a like a fluid and flexible cell membrane. Passive transport plays a primary role in the import of materials and the export of wastes. So the import of materials could be as simple as oxygen, and the export of wastes could be carbon dioxide. Now, active transport requires the direct input of energy to move molecules from regions of low concentration to regions of high concentration. Now, you may see active transport in our kidneys, for example, like as our kidneys filter our blood, we make, like as our blood is filtered, it forms this liquid called filtrate. And as that filtrate flows through our kidneys will use both passive and active transport to basically, like, if there's some kind of ion, let's pretend calcium or something that is low, as part of homeostasis, we're going to try and use active transport to like reabsorb and take that calcium back into the blood and not pee it out, like not put it in our urine. So like active transport we can use to move molecules that we need, maybe that are in a low concentration, bring them in. We also see active transport in the sodium-potassium pump, which is this GIF on the right, but I believe that's another slide coming up. So active transport is using the input of energy, so ATP, in order to move molecules opposite of their concentration gradient to where you already find a lot of these molecules. So selective permeability of membranes allows for the formation of concentration gradients of solutes across the membrane. Now this is really beautiful when it comes to our chloroplast and mitochondria in life, establishing that proton gradient that will be used to produce ATP by phosphorylation in both cellular respiration and the light reaction of photosynthesis. We also see concentration gradients of solutes in our nervous system with the sodium and potassium in their relative regions in a resting neuron. So anyway, concentration gradients can form across a membrane because those ions cannot diffuse through that lipid bilayer. Okay. And the process of endo and exocytosis require energy to move large molecules in to and out of a cell. So I want to point out, like in these two GIFs, you don't actually see ATP being used, but as you can imagine, like rearranging an entire cytoplasm would take some energy. Now on the left here, that's endocytosis. And I think endo and exocytosis are beautiful because you see the cell membrane fusing together. So on the left, it's taking, this could be like a white blood cell or an amoeba or something engulfing. And you can see how that cell membrane folds around that bacterium and brings it in. And now that vesicle is a lipid membrane, like a membrane-bound vesicle. That whole purple area is lipid bilayer. Then on the right, you can see how exocytosis can be used to send large molecules out of our cell. So this is important in the endomembrane system, where we send freshly made proteins out of the cell. So for example, if our cell, our pancreas, is releasing insulin, insulin is a protein and will be sent out of the cell in this way. And so you can see how a vesicle carrying protein will then fuse with the lipid bilayer, and then the proteins are sent out of the cell. Okay. So again, to summarize, this as kind of a repetitive concept here, you have different types of movement across the membranes. Now, for molecules that are not oxygen or carbon dioxide, molecules that are polar or charged, in order for them to move in or out of the cell, they would need to pass through protein channels. And those protein channels allow for facilitated diffusion, which is still diffusion, moving from high concentration to low concentration without the use of energy. However, facilitate means to help. So here you have a protein channel that will help the process of diffusion. So we have different kinds of facilitated diffusion. You could have like a carrier protein, which is like in C, kind of, where it helps move or carry across like specific molecules, like you would find this in the case of glucose. You could have a channel protein or an aquaporin, but they basically all allow for the movement of polar or charged molecules to cross the membrane, whether it's in or out of a cell. Okay. Membrane proteins are necessary for active transport. So how active transport is the requirement of using ATP in order to pump molecules from a low concentration to a high concentration that will require the use of ATP. And I just use the word pump. So anytime you hear the word pump or read the word pump, that is implying it's requiring energy, just like if you were to pump up a raft or a bike tire, that's going to take energy on your part. And in this standard here where it says is required for active transport of molecules and or ions across the membrane and to establish and maintain concentration gradients, that would be the case especially in our neurons, as well as our like mitochondria and chloroplasts. So here the sodium potassium pump contributes to the maintenance of the membrane potential. So the membrane potential is this difference of charge across the neuron. So if you were to measure the millivolts of like what is the difference of charge between the outside and the inside of a neuron, you would find that it's actually negative 70 millivolts. There's a slight charge, and that is due to the difference of ions across that membrane lipid bilayer. And here, when we talk about active transport, it is active transport that helps to establish that through the sodium potassium pump. So if you want to learn more about the membrane potential, because I'm not really emphasizing it in this video, I have a whole YouTube video called the action potential that would explain all of this. And I think it does a pretty good job, that video. All right, so now let's move into osmosis. I'm going to talk about the movement of water. So here, when we talk about cells placed into different concentrations of like different external environments, cells can be placed into hypotonic environments, hypertonic environments, or isotonic environments compared to their internal environment. So, for example, let's talk about like this cell here. In a hypertonic environment, there's a higher concentration of water within the cell. And therefore, the water, by diffusion, will flow through aquaporins out of the cell. The net movement, as water is moving both in and out, because it is passive transport, you're going to have a majority of water leaving the cell, resulting in the cell shriveling, which is called plasmolysis. And then, or you can see that on the GIF on the right. The water has a net movement out, which results in the cell shriveling. This is due to a high concentration of solutes in the environment. Then for isotonic, let's go there. Isotonic, which is the middle cell here in the GIF, or the cell on the right in orange. So in an isotonic environment, this is basically equilibrium. The amount of solutes on the outside or external of the cell compared to the internal environment of the cell is equal. So the net movement, water is moving into the cell at the same rate that it's moving out of the cell by diffusion. Therefore, the cell stays like the same. That is the type of environment we would prefer for our own animal cells, like for our red blood cells. Our kidneys work very hard to maintain isotonic conditions. When a person becomes dehydrated, they're losing water through sweat or not drinking, and so therefore their environment is becoming hypertonic, and therefore your kidneys will try to maintain water balance, and you probably won't urinate very much. Versus in a hypotonic environment, a human who maybe drinks way too much water has now added tons of water in its surrounding environment of its red blood cells. Now you run the risk of what's called cell lysis. In a hypotonic environment, there are more solutes within the cell, internal. So you're going to have a net movement of water into the cells, which could ultimately cause them to burst. Now for plants, let's see if I have a plant picture. Oh, I don't. But in plants, they actually will prefer a hypotonic environment so that their central vacuoles fill with water, pressing on their cell walls in order to have them stand upright. We call that turgid. Okay. In a plant cell in plasmolysis, the cell membrane will actually pull away from the cell wall and the plant will collapse if it's not like a tree with bark. Growth and homeostasis are maintained by the constant movement of molecules across membranes. So you can talk about this as far as maintaining it's called osmolarity or osmoregulation, which is like osmo, think water, and regulation, like the water concentrations in your blood. So to maintain isotonic conditions, your kidneys are under hormonal control, and the hormone ADH will actually communicate with your kidneys to regulate your urine output to maintain homeostasis. But also with growth here, I'm assuming that this standard is referencing the growth hormones or other hormones. And when it says movement of molecules across membranes, I'm wondering if they're talking about steroid hormones. So like your sex hormones like testosterone and estrogen are steroid-based hormones that can diffuse right through that lipid bilayer. And this is actually beautiful. You can see its receptor protein is intracellular, meaning inside the cell, because it is a lipid-based hormone that can diffuse right on through. So here, osmoregulation maintains water balance and allows organisms to control their internal solute composition / water potential. So part of our negative feedback loops in our body is the posterior pituitary will actually release a hormone called ADH, antidiuretic hormone, which will target the cells in your kidneys to help maintain proper water balance. So this is called osmoregulation, to help us osmoregulate. But when we talk, that's just in humans. But as far as in organisms that live in different aquatic environments, you have little single-celled organisms that will rely, like if they live in a freshwater environment, like a paramecium here in this GIF, if they live in a hypotonic environment, water will be diffused, the net movement of water will be diffusing into this cell. And as it diffuses into the cell, that cell runs the risk of bursting by cell lysis. So these little single-celled organisms have contractile vacuoles where they will actually use active transport to pump ions into this this is so beautiful. I'm going to start over. Ready? Let's focus. So they will use active transport to pump ions into this contractile vacuole space, creating a high solute concentration. Therefore, the water that has come in from the environment will now flow to that high solute concentration, or low water concentration. So it'll flow into the contractile vacuole by osmosis, and then the contractile vacuole will use ATP in order to contract and pump the water out. So think about like if you take a water balloon and you squeeze a water balloon, how the water will shoot out. It's the same idea. So that's how they osmoregulate. Whereas fish living in both marine or freshwater environments can use active transport on their gills to either, if it's a freshwater environment, they may need to pump, pump active transport, pump ions in for osmoregulation to increase the solutes within the cells. Or if it's a saltwater environment, they may use active transport to pump excess salt. When they take salt in through their food or like in through their mouths, pump that excess salt out. And then the amount of urine they produce will also be different. A freshwater fish will produce lots of dilute urine to get rid of that excess water, so their cells are not in a hypotonic environment inside their bodies. And then a marine fish will have little bit of urine, like concentrated urine, so as not to waste water. Okay. A variety of processes allow for the movement of ions and other molecules across membranes, including passive and active transport, endo and exocytosis. That's basically a nice summary slide of the different types of transport that we have talked about so far. So moving on, we're almost done, only 2 more sections. So membranes and membrane-bound organelles in eukaryotic cells, compartmentalize, intracellular metabolic processes, and specific enzymatic reactions. So basically, when we look at our different membrane-bound organelles, as well as the location of membranes, they all have their own specific jobs, and it helps to organize the different metabolic processes occurring within cells. For example, the functions of a lysosome will be very different than that of a Golgi or mitochondria. So it helps to make the cell more efficient within, so they can each do their own reactions and then not have like competing. Can you imagine if the smooth ER is producing lipids, for example, and then the lysosome breaks them down? You would want those different reactions and pathways to be isolated from each other. We've talked a bit about membranes. So internal membranes facilitate cellular processes by minimizing competing interactions and increasing surface area where reactions can occur. We'll talk more about this surface area idea in as far as inner membranes in Unit 3 when we talk about the cristae in a mitochondria. Membrane-bound organelles evolved from once living free prokaryotic cells via endosymbiosis. So let's look at these different GIFs. So here is basically the infolding of a of the cell membrane to kind of fold that rough ER and that Golgi that are all membrane bound. You can kind of see the beginning formings of a nuclear envelope around that DNA. Here it's forming like a nucleus. And then when you look at the theory of endosymbiosis, you have a prokaryote from the past, millions of years ago, being engulfed by endocytosis. So as that bacterium is engulfed, you can see how the outer foldings, like the cell membrane of this large cell, is not has become today the outer layer of the mitochondria, right? Like the outer membrane of the mitochondria. So you can see how mitochondria as well as chloroplasts have a dual membrane from endosymbiosis millions of years ago. And so when we talk about membrane-bound organelles like chloroplast and mitochondria, some evidence that they evolved by endosymbiosis is the double membrane. You can also see in the GIF on the right how mitochondria and chloroplasts will divide by binary fission. And they also have their own plasmids, similar to prokaryotes. So mitochondria and chloroplasts have their own DNA in the form of plasmids, non-nuclear DNA. They have their own ribosomes. They divide by binary fission, and they have a double membrane. And prokaryotes generally lack internal membrane-bound organelles, but have internal regions with specialized structures and functions. So while they have ribosomes, they do not have anything that is membrane-bound, like a lysosome or a Golgi or something like that. And eukaryotic cells maintain internal membranes that portion cell into regions, which we've talked about quite a bit. And that's it for Unit 2. Great job. And I hope you do well on your AP test or your unit test, whatever you're watching this video for. Okay, good job.